So earlier today, if Valeria introduced you the world of phytoplasmas and Vincent introduced to us bacteria, I will be presenting now a virus interacting with one of the interactors of a DELPHACIDE. So let's start. Before I present my study, here is the table of contents for us to have an overall view of what will my study be. For the introduction, Peregrinus maidis, or commonly known as the corn planthopper or corn DELPHACIDE, is a commercially important pest of maize. It was first recorded on corn in Hawaii by Perkins in 1982, but it is believed to have been established there as early as 1880. It is found in any place where corn is grown, but it is usually more abundant in tropical and the warmer temperate regions in the whole world. Now, P. maidis is considered to be an economically important pest of corn because it pierces the vascular tissues in the vessels of its host plant by digging through its tibial spur and sucking the sap from the leaves, leaf sheaths, and the stem of its host plant during its exploratory feeding. That physically leads to the wilting, yellowing, and in worst cases, the death of the said host plant. Besides the exploratory feeding that destroys the structural integrity of the host plant, this specific planthopper also vectors several important plant viral diseases, one of which being maize mosaic disease. Now, what is maize mosaic disease? Maize mosaic disease is from the virus, maize mosaic virus, with the species name alpha-nucleorhabdivirus maidis, and the transmission of MNV is personally to me quite interesting because MNV cannot be passed on directly from the infected plant to the healthy ones. It was also never reported to be seed-borne or disseminated in infected propagules of corn, and it cannot also be transmitted via leaf rob inoculation. Rather, MMV transmission only happens when the insect feeds from an infected plant down to the healthy ones. More specifically, the mode of transmission of maize mosaic virus is persistent, meaning it takes days to weeks for it to be passed on to the host plant, and it is retained for lifetime. It is circulative because it traverses from the foregut of the insect to the midgut down to the hindgut instead of just staying in the stylet or the gut lining of the vector. It is also said to be propagative because it replicates inside the insect vector. Side note, it is also said that the dissemination route of MNV is neurotropic, which was reported by Dr. Alvear yesterday. And this is not surprising to us because MNV does belong to the family of viruses called Rhabdiviridae, which we all know and are familiar of, similar to rabies virus of cats, dogs, bats, and humans. Now, so much for the MMV itself. We now proceed to the genome of the virus. The MMV genome is made up of a single negative sense RNA harboring 5 main core protein genes, namely number 1, nucleoprotein N, 2, phosphoprotein P, 3, matrix protein M, 4, glycoprotein G, and 5, polymerase protein L. But for the sake of this study, we turn our attention to the fourth core gene, G, that I mentioned, which is actually an integral membrane and a single spike protein that plays a viral vital role in viral attachment and fusion. To investigate the interactions directly happening in the integral membrane protein of MMVG, Dr. Karen Alvear, along with her colleagues in 2022, employed a split ubiquitin membrane-based yeast 2-hybrid system, or MBY2H system, in which they found 125 proteins in P. maidis to be interacting with the said spike protein. One corn planthopper G interactor was recovered in the MBY2H library screen. It was detected to be an antimicrobial peptide, and its name is gluginsin. Now, generally speaking, antimicrobial peptides have a wide range of activity and can effectively kill cancer cells and pathogens such as bacteria, yeast, fungi, and viruses. Now, for gluginsin, Luginsins are novel antimicrobial peptides, and this is the emphasis of the study because it was first identified just recently, very recently, last 2019, from another DELPHACIDE, niliparvata lugens. Now, why are AMPs important? AMPs are important because they are employed to intercept the growth of bacterial and fungal pathogens in post-harvest agricultural products. Now, for the management strategies of P. maidis, there are two main strategies. Number one is the use of pesticides, and number two is the development of plant host resistance. But then these two management strategies are facing problems because, as we all know, insecticide use has detrimental implications on the environment. Meanwhile, plant host resistance are faced by the problem of having limited resources for genetic resistance. So another alternative management strategies for this specific corn plant hopper is the use of biofertilizers. So the use of biofertilizers is an environmentally friendly approach and a safer alternative to reduce infestation levels and pest damage of the said insect. Now, beneficial microorganisms from the biofertilizers have been found to aid plants in combating pathogens and fending off herbivorous insects. An analysis of the luginsin expression in P. maidis with and without the supplementation of fertilizers in corn maybe could shed us some light on the indirect effects of organic agriculture, the supplementation of these fertilizers in the said certain plant-insect interaction. Now, for the objectives of the study, I want to demonstrate the potential effect of bio and organic fertilizer supplementation on plant defenses against P. maidis through the insect's performance based on PM luginsin expression. And I also wanted to investigate the differences in PM luginsin expression in both sexes, male and female, and across all fertilizer amendments in general. Now, for the materials and methods of this study, the insect culture of P. maidis was originally acquired from the adults collected from the cornfields along the Pilidride here in UP Los Baños. The colony from that specific collection was then maintained in 12 by 12 by 12 inch mylar cages and kept inside a screened enclosure at our institute. And then the horn seeds that were obtained from the Institute of Plant Breeding were grown on garden soil filled with 4 different treatments, with treatment 1 being no fertilizer or NF, natural biofertilizer being NB, synthetic organic and plus synthetic inorganic fertilizer being SO plus SI, and synthetic organic and natural biofertilizer SO plus NB. Now, for the total RNA extraction, the total RNA was recovered from P. maidis adult samples using RELIA Prep PM RNA mini prep systems following just the protocol for extracting the fibrous animal tissue. And after which, the concentration of the RNA isolates was determined using UV spectrophotometry, more specifically an epoch microplate spectrophotometry. Now, for the cDNA synthesis first strand, it was conducted using a Vivatis cDNA synthesis kit, also just following the protocol advised by the manufacturer. Now, after that, I also performed primer designing. So, primer specific to PM Lugensyn for the semi-quantitative PCR were designed using NCBI Primer Blast tool. It is an open access online bioinformatics tool. And for the semi-quantitative PCR part, the gene expression of PM Lugensyn was measured using a two-step semi-quantitative RT-PCR kit by Vivatis once more, and using the SimpliAmp thermal cycler. While for the quantification of Lugensyn, the amplicons were subjected to electrophoresis using this small electrophoresis, and then it was followed by the digital capturing process through the bio-based documentation, gel documentation system, and the gels were analyzed using a gel pro analyzer. Now, the evaluation of the expression of Lugensyn in P. maidis was compared to the evaluation of the expression of the reference gene beta-tubulin in the same insect sample. This correction was done just to normalize the possible variation in the sample. Now, for the results and discussion of my study, this is just a representative of the gels that were the results of those methods. And now for the statistical analysis, here are the results and the figure. So as we can see here, the descriptions, despite having the same purpose when they are applied in the soil, different fertilizers have different impacts on host plants and insect pests. So as you can see here, PM Lugensyn in the right side of the screen, PM Lugensyn cDNA was found to be 7 to 23 times higher in insects that fed on corn plants that were grown without the fertilizers or the controlled treatment. So for other studies have also mentioned and has observed this phenomenon wherein the combination of biofertilizer and organic fertilizer provides adequate nutrients for soil microorganisms. And the literature is also saying that the immune defense of plants is altered by arbuscular mycorrhizal fungi rather than just nutritional provisioning alone, as more than 80% of all plant species are obligate mycorrhizal symbionts. Now, if we look into the sexes of the P. maidis adult samples, we can see here that sexual dimorphism in terms of immune function is prevalent across different biological organisms. As you can see here, it is consistently expressed that the expression of PM Lugensyn in females is much higher than that of males across all fertilizer treatments. Now, literature is also saying that it is presumed that female insects possess a more robust immune system than males. And according to the statistical analyses that were used, namely the independent t-test, the Mann-Whitney test, and other post hoc tests, even though they did not have any statistically significant differences between the two sexes, we can observe that the gene expression of PM Lugensyn is consistently higher in females when compared to that of males across all fertilizer treatments. So that phenomenon can be explained by the fact that, as we said earlier, the immune function may be a reflection of an evolutionary life strategy. So this increased investment in immunity of females through extended lifespans is crucial to maximize their LRS, as they are the ones mainly responsible in the nutritional and health status of their progeny during their gravidity. Now, for the summary and conclusion of my study, PM Lugensyn was found to be 7 to 23 times higher in insects that fed on corn plants that were grown without fertilizers or the control treatment. And as we saw, female individuals consistently had higher AMP expression than males, and preliminary results seem to suggest that AMP Lugensyn selenium levels in P. maidis may be affected by the fertilizer treatment of the corn plant. So for the recommendations for research, future research outcomes, it is highly recommended that a larger sample size must be utilized since statistical power is positively correlated with the said parameter, and it would also be nice if MMV-infected P. maidis individuals will be used for further studies in investigating if a similar expression pattern of flugensin will be observed, because the test insects that I used were not tested if they were positive for the MMV, for the virus. And lastly, for the recommendations for the future endeavors of this research, it would also be helpful to analyze the expression of PM flugensin across all its developmental stages, because as I mentioned earlier, the insect samples that I used were only adults. I did not test the nymphal stages of P. maidis. So these are the references that I used. For the acknowledgements, I would like to thank Life Bank Microfinance Foundation and UST NRCP for funding my study, and I would also like to acknowledge my organizations, UP League of Agricultural Chemistry Students and UP Entomological Society, along with my two research groups, Recreational Research Group and Insect Physiology and Plant Virology Laboratory. That will be all. Thank you.